Cells are the building blocks that make up all the tissues and organs within our body. Cells need to reproduce for these tissues to grow. They do this via a process called mitosis, where the DNA in the cell is duplicated before the cell divides into two genetically identical daughter cells that have the same DNA as the original. Mitosis occurs in 4 stages: prophase, metaphase, anaphase, and of course telophase. You can remember using this acronym PMAT. Before prophase begins, the DNA in the cell is duplicated so that there is an original set and a copy. One of these genetic sets will end up in each of the 2 daughter cells that are created. Copies of DNA begin to change shape. The DNA in the cell, which normally sits in the cell nucleus in loose coils called chromatin, begin to condense into tightly coiled bundles known as chromosomes. Because the DNA has been copied, there will be an identical pair of each chromosome that join in the middle place called the centromere. During prophase, the centrioles in the cell move apart from each other and the spindle membranes begin to form between them. The next stage is metaphase. The chromosome pairs now begin to line up along the central axis of the spindle fibers between the 2 centrioles. The new and the old DNA is mixed randomly so that each new cell will have parts of the original and copied genetic material. This is to increase the chances of genetic diversity and prevent harmful mutations. During anaphase, the separation of the DNA begins. The spindles retract away from the central axis towards the centrioles, pulling the chromosome pairs in half. In this way, half the chromosomes are pulled to one side and half go the other way. The last stage is telophase. Now that the chromosome pairs have been separated, the cell membrane begins to close in and split the cell into 2. When this stage ends, there are now 2 separate cells, each with an original set of DNA.